a disease in plant. And this disease is called the Crohn gall. And indeed, the bacteria is making plant cell proliferate, and it forms tumors that you can see here. Okay, and to induce those tumors, the bacteria is able to inject a small piece of DNA within the plant cell. And this small piece of DNA will be inserted into the plant genome. And then it will be expressed in the plant and so on. And I told you that this small piece of DNA in the bacteria is carried by a plasmid that we called the tumor-inducing plasmid, or TI plasmid, tumor-inducing plasmid. And I introduced you with the structure of the plasmid, and I told you there is this tDNA part. It's called tDNA because it's a part of bacterial DNA that will be transferred to the plant cell. And you have a region which is called the virulence region. It's responsible for the virulence of the bacteria, and it carries several genes. I told you on the tDNA, we could find 3 genes. 2 genes are responsible for the biosynthesis of phytohormones. And the transformed plant cell will be able to synthesize auxin and cytokinin. And we spoke about those phytohormones during the part of the course on in vitro culture. When you have an equal balance of those 2 phytohormones, it induces the cell to divide. So you will have a proliferation of the plant cell, which will form the tumor. Then the tumor will produce opines, and opines, it's a metabolite that the bacteria will use for feeding. Okay, so we will kind of shortly see how those different Vir genes are used. How does the bacteria infect the plant and transfer this tDNA part into the plant genome? Again, if you have any question, if I need to stop to repeat something, you tell me. Huh? So what you need to know is that a bacteria will be able to infect the plant when the plant is wounded. When a plant is wounded, it will produce some chemicals, some phenolic compounds. It will produce compounds that will attract the bacteria. Okay, and these are the chemicals that are produced by the wounded plant, you see, and those molecules are mainly acetosyringones. And this acetosyringone will be perceived by a receptor at the surface of the bacteria. And this receptor is encoded by one of the Vir genes. It's Vir-A. So a chemical produced by a wounded plant will be recognized by Vir-A, and this will induce a cascade, and all the other Vir genes will be expressed. So finally, when the bacteria perceives a plant which is wounded, it will induce its virulence. Okay, the bacteria will become virulent because of the expression of the Vir genes. Those Vir genes, they are all carried by an operon. It's what we call the Vir operon. Okay, so when the receptor encoded by Vir-A perceive acetylsyrangone, all the other genes will be expressed. Okay, and all those Vir genes will have different functions during the interaction between the plant and the bacteria. Okay, Vir-A, and together with Vir-G indeed, is involved in perception of the plant and activation of the virulence. Other genes will be responsible for the transfer of the tDNA into the plant cell nucleus, and for its insertion into the plant genome. And several Vir genes encode for protein, which will constitute what we call a type 4 secretion system. I will explain you what it is. This is the type 4 expression system. You see that it's made of Vir proteins, and those Vir proteins will assemble to form a syringe. And you see it here in green. This is the inside of the bacteria, and the envelope of the bacteria is here, and it will form a needle that you can see here. And this needle will be used to pierce the plant cell wall and to inject the tDNA into the plant cell, which is here. So several Vir genes encode for proteins. You see Vir-D4, Vir-B4, Vir-B11, B8, B6, B10, B7, B9, B2, B5. All those Vir proteins will assemble to form a needle necessary to pierce the plant cell wall and to inject the tDNA inside the plant cell. Then some other Vir proteins are necessary for the injection of the plant cell. So you see here the type 4 secretion system, the syringe, which is made of Vir protein. Another protein, Vir-D2, will be necessary to excise the tDNA. It will cut the tDNA, and it will help to transfer the tDNA through the type 4 secretion system inside the plant cell. And then it will help the tDNA to move towards the nucleus. The protein Vir-D2 will also help to import the tDNA inside the nucleus, and there it will help to insert the tDNA into the plant genome. Another protein is very, very important for the injection of the tDNA. It's the protein Vir-E2. It's this pink protein. And this pink protein, Vir-E2, will coat the DNA. It will cover the DNA, and in fact, it will protect the tDNA from the destruction by nucleases. Okay, nucleases, it's enzymes that are able to cut, to digest the DNA, and to protect the bacterial tDNA. It is covered, protected by the Vir-E2 protein. Okay, and you can recognize the Vir-D2 protein that help to guide the tDNA toward the nucleus. Finally, inside the nucleus, you will have action of two other protein, Vir-F, and another plant protein, VBF, that are involved into the integration of the tDNA. It's Vir-F, which is responsible for the integration of the tDNA into the plant nuclear DNA. Okay? what you must understand is that the Vir protein, they are not working alone. To be able to transfer, to guide the tDNA towards the nucleus, to be able to make the tDNA enter into the nucleus, to be able to integrate the tDNA into the plant genome, the VIR-E2, VIR-D2, and VIR-F proteins will interact with plant proteins that are key actors of the interaction. Okay, there are a lot of plant protein that will help for this process. The VIR-E2, VIR-D2, and VIR-F protein alone will not be able to help the integration of the tDNA into the plant genome. Those bacterial protein, VIR-E2, VIR-D2, and VIR-F, will interact with a lot of plant protein. So in blue here, it's all the plant protein, the host protein, which are necessary for the integration of the bacterial tDNA into the plant genome. So VIR-D2, VIR-E2, VIR-F, and some other VIR protein will manipulate some plant function for helping the integration of the tDNA into the plant genome. Okay, this is a kind of summary. When a plant cell is hurt, wounded, it produces phenolic compounds, acetylcyrandoate. And this is perceived by a receptor, which is the VIR-A protein. The VIR-A protein will phosphorylate the VIR-J protein that will induce the expression of VIR proteins. Okay? Then VIR-D2 will help to cut the tDNA, will help to extract the tDNA. VIR-D2 together with VIR-E2, E3, and F will help to guide the tDNA through the type 4 secretion system, which is made of VIR proteins. Inside the plant cell, VIR-E2, VIR-D2, VIR-F, and some other VIR protein will help to guide the tDNA towards the nucleus, and it will help for the integration of the tDNA into the plant genome. There is one very, very, very important point to remember, is that the insertion of the tDNA into the plant genome is completely random. The tDNA may be inserted anywhere into the plant genome. It can be inserted anywhere, it's completely random. So this is how works the infection process. Once the tDNA is integrated into the plant genome, it will be expressed as a plant gene. The same, it will be considered as a plant gene, and thus it will be expressed. And of course, it will allow the expression of auxin and cytokinin. The plant cell will proliferate, and it will produce opines from which the bacteria will feed. This is how Agrobacterium tumefaciens is able to infect plant cell, to manipulate plant cell physiology, to feed from those plant cells. Now, when you generate a GMO plant, you will use all this property of Agrobacterium tumefaciens. You will use the property of Agrobacterium tumefaciens to insert its tDNA into the plant cell nucleus. But what you will do is that you will replace the genes that are naturally present on the TI plasmid in the tDNA. You will replace them by your gene of interest. And of course, you will want to be able to select the plant cells that will be transformed, so that will have acquired the tDNA. So you will also insert into the tDNA a selectable marker. And that's all. That's the principle of plant transformation using the bacteria Agrobacterium tumefaciens. Because here the tDNA is not anymore carrying the gene responsible for plant cell proliferation, you will not anymore have formation of a tumor. You will not have any more production of opine, but anyway, the bacteria will have been able to insert this tDNA into the plant genomic DNA. Did you all understand? Did you all understood the principle of making a GMO plant using Agrobacterium tumefaciens? It's a very important part of the course. It's all resumed on this slide. Do you have any question on this? Do I need to repeat something, or is it clear for everybody? Do not hesitate.
What T4SS stands for?
Sorry? The T4SS, I told you, it's the type 4 secretion system. Type 4 secretion system. So you understand that it will help the bacteria to secrete the tDNA and some proteins into the plant cell. It's a secretion system. In bacteria, you have several kind of secretion system. I think now 7 types of secretion systems have been identified in bacteria. The type 4 secretion system is kind of typical from Agrobacteria, and it's responsible for the transfer of the tDNA and some bacterial protein into the host cell.
Okay, thank you.
Do not hesitate if you have other questions.
I have a question.
Yes.
Is auxin and cytokinin is naturally in the plasmid? Are they naturally?
Yes. So these are genes. It's not the auxin and the cytokinin, which are naturally present on the TI plasmid. It's genes that will allow the expression of enzymes that are necessary for the biosynthesis of auxin and cytokinin. So once those genes are inserted into the plant genome, you will have in the plant cell expression of the genes which will allow the production of auxin and cytokinin. So on the tDNA, it's genes that are present. Genes always encode for proteins, and the proteins that will be produced will be responsible for the biosynthesis of auxin and the biosynthesis of cytokinin. Okay?
Okay, I understand.
You understand? Perfect.
Yeah.
Other questions? It's clear for everybody?
Yes, I don't have any question for now.
Yeah. So I continue then. But you see, do not hesitate. If something is not clear, you ask. So now you know the principle. What you need to understand is that for a gene to be expressed, for a gene to function, it needs, of course, to have a coding region, what we call the open reading frame. It's really the DNA sequence that will encode for a protein. And then you know that in the structure of a gene, you have some UTR, the 5' UTR and the 3' UTR, which are untranslated regions. So this will not be translated into protein, but they are necessary region for the expression of the gene. It's regulatory sequence that will allow correct expression, correct production of the protein. And then you have upstream of the 5' UTR a promoter, which will regulate the expression of the gene. So this is the structure of a eukaryotic gene. So of course, if you want a gene to be expressed into a plant cell, it will need to have this structure. So finally, the gene that you will insert here and the gene encoding for the selectable marker, they will need to have this structure, and they will need to have regulatory regions that are functioning into a plant cell. Okay? They will need to have regulatory regions that are working properly into plants. Okay? It's very important to remember what is the structure of a gene. You have, of course, the open reading frame, which is encoding for the protein. Then you have the 5' and 3' UTR, untranslated region. Those are regulatory regions that will allow the production of the protein. And then upstream of the 5' UTR, you have a promoter, which will regulate the level of expression of the gene. So the gene you will insert into a plant cell when you will make a GMO, it will need to have this structure to be functional into a plant cell. Then we will see how does look like a tDNA region that we use for making a GMO. So I just explained you that the gene of interest, it's this one. If you want it to be expressed in a plant, it will need to be recognized by the plant molecular machinery. It is very important that it's recognized by the plant machinery to be expressed. And for the gene to be recognized by the plant molecular machinery, it has to have those regulatory regions. It's necessary. So this is a typical example of a tDNA you will make to express into a plant gene, a gene of interest. What can you recognize on such construct? The left border and the right border, LB and RB, or the, I come back, it's the bording region you see here in yellow, and they will always be present. They will be necessary for the transfer of the tDNA. So when you generate a construct to produce GMO, you will always have those borders, and they are called the left border and the right border. In this tDNA, we can recognize two genes. One, it's HPT, and the second gene is AFP. AFP, it's the gene of interest. It's the gene we want to express into the transgenic plant. The HPT gene will allow the selection of the transformed plant. It's a gene that will allow the transgenic plant to resist to an antibiotic. So we will be able to use this gene to select the genetically modified plant cell. And you see that this gene and this gene of interest are both surrounded by two regulatory regions. This, and this, and this. You can recognize a promoter, and you will need a promoter region working in plant to drive the expression of your transgene. And here, the promoter which is used, it's a very strong and what we call a constitutive promoter. It's the P35S. You see it here. The P35S, it's a promoter of a virus gene, whatever. In a plant cell, it will, it will allow the constitutive expression of the transgene. What does it mean, the constitutive expression? It means that the gene, HPT and AFP1, will be expressed in the transgenic plant, in all cells, in all tissue, whenever the developmental stage of the plant. It's constantly expressed in all plant cells at a high level of expression. So that's what we call a strong promoter. It will allow a constitutive expression of the transgene. You may use some other kind of promoter. You may use inducible promoter. Inducible promoter, as their name say, you will be able to drive the expression of the transgene because the expression will occur only under certain conditions. You may use a compound, a chemical compound, to induce the expression of the transgene. For example, you can use alcohol. There are some ethanol inducible promoter, and then the gene of interest will be expressed only when you expose the transgenic plant to ethanol. You have estrogen inducible promoters that are allowing expression of the transgene only when you expose the transgenic plant to estradiol. You have different kind of inducible promoter. You have some heat inducible promoter. It means that you will induce the expression of the transgene only if you expose the plant to high temperature. There are some cold inducible promoter that will allow to induce the expression of the transgene only when you expose the transgenic plant to cold temperature. So all those kind of promoters are what we call inducible promoters. The use of inducible promoter allows to switch on the expression of the transgene whenever you want. Okay? Otherwise, the promoter is not working, you don't have expression of the transgene. If you expose the plant to a stress, or if you add a chemical to water the plant, it will switch on the activity of the promoter and the gene will be expressed. You may use a third kind of promoter, is that we call specific promoter. It's a promoter that will allow to specifically drive the expression of the transgene. I don't know, for example, in one plant tissue, you may want the gene to be expressed only in the flowers, or only in the roots, only in the fruit. You can choose in which tissue you will express the gene. I give you an example. You want to improve the quality of tomato, and you identify one gene which is responsible for the quality of the tomato fruit. You may not need to express this gene in all the tissue of the tomato. You don't need to have expression of the gene in the root, neither in the leaves, because maybe it will affect the plant development. So you want to drive your expression specifically into the fruit. So you will choose a fruit-specific promoter. Okay, so this is for the choice of the promoter region. And on this small scheme, sorry, remember here is the left border and the promoter here and here. This promoter is upstream of the HPT gene, and this promoter is upstream of the FP1 gene. Second region of the tDNA is the coding regions. And here there are two, because we have two genes on the tDNA, one gene which is HPT, a second one which is FP1. So you need the coding region. Take care, the structure of one eukaryotic gene, very often you have non-coding region separating coding region, is what we call the intron. And remember, the intron, which will be separated by two exons, they will not be translated into amino acids. So normally they are removed during one important state of the transcription process, which is the splicing. When you generate a tDNA construct, you use only the coding sequence, the coding region of a gene. So you don't take into consideration the introns. You work with a cDNA sequence. Huh? This is very important to remember. You insert only the coding region of the gene of interest. And it's this one and this one. And then you need a termination sequence with a polyadenylation region to generate some messenger RNA. Remember, in eukaryotic cells, the messenger RNA, mRNA, is polyadenylated, so you need a terminator. And it's almost always the same. It's a terminator of one gene, which is encoding for the nopalin synthase. We call it the TNOS terminator. And you can see it here after this gene and here after this gene, the TNOS. The TNOS terminator, which is necessary to have a functional messenger RNA. So all this region will allow the constitutive expression of the HPT gene, and all this region will allow the expression of the AFP1 gene. Okay, remember this, huh? You will need to have regulatory region into your tDNA for the gene to be functional into the plant cells. And depending what you want to achieve, you will use different regulatory region. You understood that the choice of the promoter is very important. Depending on what you want to do, you will choose different kind of promoters.
Can I ask something?
Yes.
Okay, so first of all, as I understand, if I understand correctly, then each ORF
Yes.
open reading frame needs at least one promoter, right?
Yes.
Okay, and secondly, the termination sequence belongs it belongs to the UTR region, or
if you can consider that it's the 3' UTR. It's not translated, and it's necessary for regulating the transcription. So it's part of the 3' UTR.
Okay, I understand. Thank you.
And similarly, in the construction you make, you will have very often it's included in the promoter. You have the 5' UTR.
Yes, so the 5' UTR, it's in between the
promoter and the
P50S.
open reading frame. Yes.
Yeah. Okay. I understand.
Other questions? So I continue. There is probably something I didn't tell you. So what you need to understand, if you want, sorry, to transform a plant, if you want to have this tDNA, to use such a tDNA to transform a plant, you will need to build this construct. You will need to generate, to engineer a plasmid, a TI plasmid, and you will choose what you want to put into the tDNA. And to build this TI plasmid, you will use molecular biology tools. It's what we call cloning. You will do cloning. You will need to amplify some DNA sequence doing PCR. You will need to insert those DNA sequence into the plasmid to step by step build such a construct. Okay? You will need to build this DNA sequence. So for this, you will use molecular biology tools. And you know that when you do cloning, for example, you cut plasmid, you ligate plasmid, and to multiply the plasmid, to amplify it, you use a bacteria which is Escherichia coli. It's the bacteria which is used to do molecular biology to manipulate DNA sequence. Okay, so it's very important that you consider this. Huh? We are speaking about generating a new kind of TI plasmid to generate one specific GMO plant. This, you will need to build it, I would say, with your hands. You will do a lot of pipetting. It's something that you do on the bench. It's molecular biology. And this cloning, this gene cloning, you do using the bacteria Escherichia coli. What you need to understand is that the TI plasmid of Agrobacterium tumefaciens, it's a huge plasmid. It's a very, very big plasmid. So when you do molecular biology in order to generate GMO plants, we don't work with a full TI plasmid because it's too big. So we will use some specific strain of Agrobacterium tumefaciens that will carry two plasmids. They will be what we call a helper plasmid, on which you will have all the vir genes necessary, you remember, for the virulence of Agrobacterium. And what you will manipulate at the bench to generate your tDNA, it's only a small binary vector. It's much smaller, and it will be much easier to manipulate with your molecular biology tools. Okay? It's called a binary vector because it's able to replicate in two kinds of bacteria. It's able to replicate in Escherichia coli, and this will be necessary for the building of this vector, and it will also have the same plasmid will also be able to replicate into Agrobacterium tumefaciens. And you remember, it's Agrobacterium tumefaciens that we will use to generate the GMO plant, to transform the plant. But before to be able to insert into the plant your gene of interest, you need to insert this gene of interest into a tDNA. So you will do molecular biology, you will construct de novo a binary vector that will be used then to generate the GMO plants. Okay, so you have to understand this principle, the construct you use, the tDNA you will use to generate your GMO plant is not coming from nowhere. You will need to construct with your hand this tDNA, depending on what you want to do. Okay, so you manipulate in the laboratory a binary vector that you will then introduce into Agrobacterium tumefaciens, and then you will use this Agrobacterium tumefaciens to transform the plants, to make the GMO plants. Okay, I will come back on this region of the tDNA. I told you that this region was necessary to be able to select the cell that will be genetically modified. And I told you that this gene will allow the transformed cell to resist to an antibiotic. That's what we call the resistance gene. And when you generate transgenic plant, you will need always to have a marker, something to be able to select the cell that will be transformed by Agrobacterium tumefaciens. And there are different kinds of selectable marker. Okay, selection gene. I open a bracket, look at the structure of this tDNA. Here you have the left border, LB. Here you have the right border, RB. You can recognize promoters. The promoters are upstream of the gene of interest. Okay, here there is a promoter, it's driving the expression of this gene. And then, of course, after the gene of interest, you have a terminator, the TNOS. You notice the orientation of the construct, left, right. Before, we had something opposite. You see, we had left and right on the other side, and of course, the promoter were upstream of the gene. You have the arrow, which is guiding you to identify the orientation of the construct. It's going on this side, left, promoter, gene, terminator, promoter, gene, terminator, right. Here, it's the other way around, left, promoter, gene, terminator, promoter, gene, terminator, right. Okay. The selection genes, they may be of different kind. I was speaking before of the HPP gene here. The HPP is encoding for hygromycin phosphotransferase. It will allow the transgenic plant to resist to an antibiotic, which is hygromycin. You may use the NPT2 gene, and those one are the most commonly used selection genes. The NPT2 gene, it's encoding for neomycin phosphotransferase of type 2. It will allow the transgenic plant to resist to another antibiotic, which is kanamycin or neomycin, it's the same one. You all know that antibiotics are normally acting on bacteria. They are or bactericide, they kill bacteria, or they are bacteriostatic. They stop the growth, the multiplication of the bacteria. Antibiotics can also be used as herbicides. Not all of them, but some of them. Those ones can be used as herbicide because it's antibiotics that will inhibit the protein synthesis, and they are active also on plant cells. So they will inhibit the synthesis of the protein into the plant cell, so the plant cell will die. You may also use as a selection marker true gene allowing resistance to herbicides, to true herbicides. For example, the EPSPS gene, which is encoding for an enzyme which is called enol-pyruvate phosphate synthase, it will allow the transgenic plant to resist one very famous herbicide, which is glyphosate. Glyphosate, it's the same as Roundup. I don't know if you know this name. It's the most famous herbicide, which was produced by Monsanto. Okay, so remember, you will need to select the plant cells that have been transformed by Agrobacterium tumefaciens. So the plant cells that have been transformed, they will be able to express selection gene, and this selection gene will allow resistance to an antibiotic or an herbicide. So what you will do after you have transformed the plant tissue, you will grow this plant tissue in vitro, during in vitro culture, into a media that contains the antibiotic or the herbicide, and only the plant cell in which the cDNA has been integrated will be able to survive. It's necessary to select the transgenic plant or transgenic plants. So what is the usual procedure to make GMO plants with Agrobacterium tumefaciens? It can be used with a lot of different plant species. Most of the plants, more than 60% of the plant species, can be transformed with Agrobacterium tumefaciens this way. You will take a plant, you take some explants, so some plant tissues, and normally it will be leaf discs. But it can also be some small pieces of stem. It can be embryo. It can be different kind of tissue, but most of the time it's a small leaf disc. And you will put those leaf discs. So it's in vitro culture. You understand that you work in vitro. So you work under sterile condition. You must not contaminate your plant tissue. You will grow your leaf disc on a petri dish, and on the same petri dish you will put your agrobacteria. It's a step which is called the co-cultivation step. You will co-cultivate together the plant tissue and the bacteria, Agrobacterium tumefaciens, carrying the TIA plasmid that you created. Because the bacteria is in contact with wounded plant cell, it will be virulent and it will insert a tDNA in different cells. Okay, but what you need to understand is that this little disc is made of thousands of cells. Only very, very few cells will be transformed. And for example, you see on this little disc, finally there are only 2 cells that have been transformed. On this little disc, only 1. On this one, only 2. To select the genetically modified cell, you will grow the tissue on a medium which contains, for example, the antibiotic you use to do the selection. So here you move from the, let's say, infection inoculation media to the selection media. And in this media, you will put suitable phytohormones in addition to the antibiotic or to the herbicide. You will use phytohormones to make the transgenic cell proliferate. So one cell which is resistant, for example, to the antibiotic, is able to proliferate, to divide, to form a callus. So you will get like this calli, which are made of clones of transgenic cells that will all have the same genotype. But it's very important what I will say here. Remember, when Agrobacterium inserts its tDNA into the plant genome, I told you it's completely random. The tDNA can be inserted anywhere. So there is no chance that this cell will have integrated the tDNA in the same locus as this one. Okay? Similarly, this one will not have integrated the tDNA at the same locus into the plant genomic DNA. And it's the same for this one, it's the same for this one. So from this experiment, you will regenerate 1, 2, 3, 4, 5 transgenic plants that will all have a different genotype because they will not carry the tDNA at the same locus into the genetic DNA. Clear? And then you use again in vitro culture to regenerate from the calli whole transgenic plant. So you see that to make GMO plants, you need to use in vitro culture. And you remember, depending on the plant species, it may be tricky to cultivate, to regenerate a whole plant from a plant tissue. So this is really a long and tedious experiment with this tobacco plant. This is a tobacco, you can recognize it as the flower. It's very easy to do, it will work easily. And you will need a few months to select transgenic plants. But depending on the species of the plant you transform, it may be more or less difficult and more or less long. Because all the plants are not similarly susceptible to Agrobacterium tumefaciens. Let's say that the efficiency of transformation is very high for tobacco, but it will be much less for other crops, for example. Okay, and then you have the state of in vitro culture to regenerate a complete transgenic plant, And this, depending on the plant species, it may take some time. So this is the protocol, the procedure to make a transgenic plant. Okay. What I want you really to understand is that it's only a few cells that will be transformed. Here it's 1, 2, 3, 4, 5 cells for 3 leaf lists that will be transformed. So you need to select the cells that have been transformed. And for this, you will use your selection gene. So you will grow your plant tissue after the co-cultivation step. You will grow your plant tissue on a medium which will allow to select the transgenic plant, and all the other cells will die or not divide. And then you will need to regenerate whole plants. You need also to remember that in those 5 transgenic plants, the tDNA will not have been inserted in the same place into the genomic DNA. Insertion will occur in different loci because it's completely random. And there is, I would say, if you do one experiment like this, there is no chance, it's not no chance, but almost no chance that you will have insertion in the same loci. So the 5 transgenic plants somehow will be different. Clear? Question on this important slide? So I continue. This is what I just explained here, is that you need to be aware that you will need a long and tedious in vitro culture step to regenerate a transgenic plant from the transformed cell. Okay, it will be long and fastidious, and you will need to keep your plants healthy to avoid any kind of contamination until you are able to harvest seeds from your transgenic plant. Okay. So a lot of in vitro culture is necessary. I want to say 1 or 2 words about another transformation method. Here it's what we call the leaf disc transformation protocol, and there is another method which is a floral dipping method, which is used almost exclusively for Arabidopsis thaliana, which is a model plant in laboratories. In all the laboratories doing functional genomic, I mean, not in all, but in most of those laboratories, researchers work on the model plant Arabidopsis thaliana, because it's a plant kind of simple with a small genome, only 5 chromosomes, very easy to grow. It takes only a few weeks to grow the plant, so you can have about 5 to 6 generations in 1 year. It's giving a lot of seeds. One plant will give hundreds of seeds, so it's a very, very interesting model. And then its genome is completely known, so it's also very important for doing this kind of work. To transform Arabidopsis thaliana, it's very easy. You take a plant, you wait that it flowers, and you wait that it's having enough flowers, and you will, I don't know how to say it in English, you will put the flowers into a suspension of agrobacteria. So this is, let's say, agrobacterium culture in a liquid medium, and you will drench your flowers into the suspension of bacteria for only a few minutes, 2 minutes. Then you let the plant continue to grow. You let the flower maturating, making fruits that we call the calyx, and you harvest the seeds. I told you in Arabidopsis plant like this, you will have hundreds of flowers. And from those hundreds of flowers, you will harvest thousands of seeds. And you will, to select the transgenic plant, you will only have to grow the seeds on media, for example, on Petri dish, containing the selectable marker. So it may be the antibiotic, and you will select the transgenic plants. So it's a very, very easy method. And what is most important, it's the seeds that will be transgenic. Insertion of the tDNA will occur or in the pollen or in the ovule, so in the germ cell. And Arabidopsis thaliana, it's autogam, so you will have autofecundation, and some seeds which will have acquired a tDNA will be producing a transgenic plant directly. So using this method, you will not have to do in vitro culture to regenerate from a plant tissue a whole plant. You will only have to collect the seeds. You put the seeds, for example, on soil, you spray with the herbicide or with the antibiotic, and only the transgenic plant will grow. So you get a kind of directly transgenic plant. Okay, I want to speak a little bit about the limitations of the Agrobacterium tumefaciens transformation. What you need to know is that Agrobacterium tumefaciens is not usable to transform any kind of plants. Some plants cannot be transformed via Agrobacterium tumefaciens. And it's true for many monocots. You know, monocot plants, it's all the gramineaceous plants, the wheat, the corn, the rice, barley, those are monocots. Those plants are maybe more difficult to transform with Agrobacterium tumefaciens. But it's not true for all of them, and a lot of progress have been made. And now it's becoming possible to transform barley using Agrobacterium tumefaciens, so also rice. A lot of progress has been made, and I would say that I should write now that transformation is not as efficient with monocots, but it's becoming more and more used anyway. But when you cannot transform one plant species with Agrobacterium tumefaciens, you can use different strains that may be more virulent, or there are anyway some alternatives to increase the efficiency of transformation using Agrobacterium tumefaciens when the plant species is recalcitrant through transformation. You may also use some other agrobacteria. Agrobacterium tumefaciens, I told you it's responsible for the disease called Crohn-Gall, sorry, and it's because Agrobacterium is able to transfer its tDNA into plant cell. You may use other agrobacteria that are also somehow able to insert a piece of DNA into a plant cell. You have, for example, Agrobacterium rhizogenes. Agrobacterium rhizogenes, it's a pathogenic bacteria which is responsible for the hairy root disease. And it will make root proliferate. It's this one. Huh? It will make root proliferate. So the plant will produce a lot of roots. but it will be roots that will be kind of strange. They will have no more geotropism, and they will be able to grow without putting hormones into a media. So the roots will proliferate. And this is because Agrobacterium rhizogenes is able to insert into the plant cell not a tDNA, but what we call a rDNA. You have another strain of Agrobacteria which is able to transform plant cell, but which is less used because it's not infecting so many plants. It's Agrobacterium rubi, and it's also making galls, but not the same galls as the crown gall. It makes aerial galls that are much smaller, only in some kind of plants. I just say one or two words on Agrobacterium rhizogenes because it's used a lot to make airy roots. We know now that Agrobacterium rhizogenes is able to transfer into a plant cell a large plasmid, which is called the RI plasmid for root-inducing plasmid. It's somehow working similarly to the TI plasmid. You have some virulence gene. You have RE plasmid carrying a tDNA region that will be transferred to the plant cell, but still how it works, it's not fully understood yet. Okay, it's not a model which is as known as the TI plasmid, but it's working kind of similarly. It's used a lot in biotechnology because you may use those airy roots produced at an industrial scale some metabolites. And it has been used to produce chemicals, medicines by industrials. Okay? So remember, Agrobacterium rhizogenes, you can use it to transfer a gene of interest into a plant cell. It will allow root proliferation, and you may use those roots produce metabolites in the industry. Okay, this is the principle of plant transformation. Do you have any question on this part of the course? Really, do not hesitate. Did you all understood, particularly, did you all particularly understood what I was speaking about here? And here. Did you understood all the principle of plant transformation using Agrobacterium tumefaciens? Do you have any kind of question on this part of the lesson? Do not hesitate. If you have question, ask. If you don't have any question, we will continue.
I have a question.
Yes, please.
For the selection medium, can you explain why we use an antibiotic to select
the transgenic plant?
Yeah.
Yes.
But I understand that well, the antibiotic will affect on the bacteria, but I don't know how it can kill the plant cell.
Yeah,
only the transgenic cell can grow.
Yes, I will explain you now. Okay. So I come back to this, and it's a very important point. When you will generate your tDNA, I told you, when you transform a plant tissue, the agrobacteria is able to transfer some plant cell, but it's not so efficient. Huh? It will not be able to transform 100% of the cells. It will only be able to insert its tDNA in a few cells. And in this example, I told you we used 3 leaf discs. Each leaf disc is having thousands of cells, but finally at the end, only 1, 2, 3, 4, 5 plant cells will be transformed. You need to be able to select them. If you make, you put directly the leaf disc on a media that allow plant cell to proliferate, but you don't put, you don't select the transformed cell, all the cell will multiply and you will get huge callus, but you will have most of the plants that you will regenerate from those callus that will not be transformed. So you need to select specifically the plant cell in which a tDNA has been inserted. To be able to select those plant cells, I told you that we use selection genes. And selection genes will allow the plant that have been the plant cells that have been transformed, to resist to an antibiotic or an herbicide. Why we use antibiotics that are normally, it's true, acting on bacteria? It's because those 2 specific antibiotics are inhibitors of the protein synthesis. And those molecules are able to inhibit the protein synthesis not only in bacteria, but also in plants. So finally, when you will treat a plant with kanamycin, hygromycin, it will not be anymore able to produce protein, so it will die. We put the genes encoding for those enzymes into the tDNA. So the plant that will have integrated the tDNA, the transgenic plant, they will produce this gene. And this gene is able to degrade the antibiotic. It's able to inactivate the antibiotic. Okay, maybe it's here that I was not clear. Huh? Those enzymes, they are made to make the plant resistant to the antibiotic. So finally, the neomycin phosphotransferase is inactivating the neomycin, the kanamycin. The hygromycin phosphotransferase is able to inactivate the hygromycin. And the EPSPS is able to inactivate the glyphosate. So the plant that will integrate this tDNA, they will be no more susceptible to those chemicals. They will resist those chemicals. So here, if Agrobacterium tumefaciens is inserting here its tDNA in this cell, when you will grow this cell on the selection medium containing the antibiotic or the herbicide, it will be able to inactivate this antibiotic, so it will survive. All the other cells, they are susceptible to the antibiotic, they will not be able to produce protein anymore, they will die. Only the plant cell carrying the selection gene will survive when you add the antibiotic or the herbicide to the selection medium. So like this, you can select plants that have integrated the tDNA. Okay? So I told you, you may use antibiotics or herbicide because all of them are killing plant cells. If you insert into the plant cell, into the genome of the plant cell, those genes, they will be able to resist those chemicals. Did you understand better now? Is it more clear?
Yes, I understand.
Okay, perfect. Do we have some other questions? Yes.
Can I have a question? So after you select the cell, so it go to like 5 colors. So in that slide, it will be like the relation to say that atokinin and cytokinin is responsible for like shoot grow or root grow.
Yes.
So in after the selection, the next slide will have like higher cytokinin, right?
Yes, yes, exactly. You understood perfectly. After it's in vitro culture, so you need to follow all the necessary process to regenerate a whole plant when you use in vitro culture. So first, you use a media that will allow to select transformed cells and to make those plant cells proliferate. And then you will use a shoot, for example, a shoot-inducing medium. So you put cytokinin, it will make shoot. You transfer to a medium that will allow growth of the shoot and the root and so on. So you will drive the organogenesis to be able to regenerate a whole plant. That's why I told you it's not a very simple method. Huh? You need to respect all the in vitro culture procedure to regenerate from one single transformed cell a whole plant. So you will need to change the media and so on to first select the transformed cell, make it proliferate. Then you will need to make shoot coming from this callus. Then you need to regenerate a plant with roots and so on. So you need to use the complete in vitro culture procedure to be able to regenerate from one plant cell a whole plant.
Okay, thank you.
Other questions? Okay, so we will make a break. It's 9:27. Okay, we take a 13-minute break. So we are back. I don't know what time is it for you, but we are back at maybe for you it's 3. So we are back.
We'll be back by 3:40.
At 3:40.